Good morning or good afternoon, everyone, and welcome to the bonus lecture on our immunity. So, this is a lecture provided for our students so they may better understand how immunity works, especially with our medical laboratory students, in which they will have a future course of immunology. This may be a good backbone for their prerequisite knowledge. So, let's start. So before we begin, I would like to post a notice that this lecture will reference the anime Cells at Work or Hatarako Saibo. You are highly encouraged to watch the anime after finishing this lecture, as you will better appreciate the scientific principles behind the anime and what the anime represents. So let's start with immunity. We have two types of immunity. We have innate, which is characterized by a general and rapid response, and we have adaptive. It is specific to each pathogen, but it is relatively slow compared to our innate immunity. So we have these chemicals called cytokines. These are proteins or peptides that bind to receptors on cells to stimulate a response. They can be self targeted, meaning they can target themselves. Later, you will see an example of a self targeting cytokine. So once a immune cell detects the presence of a specific cytokine, a response can include either attraction of other immune cells, promote phagocytosis or eating the pathogen, stimulation and proliferation and differentiation of our immune cells, and also it can regulate intensity and duration of the immune response. We have different types of cytokines, each with its own source and its own purpose. First, we have our interleukin that comes from our leukocytes. Leukin, leuko. We have our lymphokin that comes from our lymphocytes. We have our chemokine that promotes chemotaxis or the movement of cells. We also have interferon. This interferes with our viral replication. And we have our inflammatory cytokine, which promotes inflammation. So let's meet an important term, which is our antigen. So, antigen is a substance that binds to receptors on adaptive immune cells to stimulate an adaptive response. We have two types. It can be foreign, meaning it is formed and in outside the body and can enter the body through various ways, and we can also have self antigens. So self antigens, you can consider this as relatively non-immunity producing substance. However, if you have an abnormality in your immune system, it can provoke the your immune system to respond. So self antigens are molecules produced by the body that can stimulate an immune response. So we have good sides and bad sides. For our beneficial, it can recognize tumor antigens and subsequent destruction of those tumor cells. For the harmful effects are our autoimmune diseases, such as our rheumatoid arthritis. So we have different types of our immune cells. We have B and T cells. These are our primary adaptive immune cells. So these are lymphocytes, meaning they come from the lymph, that can bind antigens. When B cells are activated, it eventually creates antibodies. Antibodies are proteins that can bind to the antigens and stimulate their production. T cells, on the other hand, when T cells are activated, they can produce both cytotoxic T cells and helper T cells. We will discuss the effects of cytotoxic T cells and helper cells later. So let's discuss first the cells under our innate immunity. So when we say innate immunity, this is the general response. So some terms we need to discuss first is our PAMPs or pathogen associated molecular patterns. So these are patterns of or an arrangement of proteins These are molecules common to pathogens but are not found in human cells. An example are parts of bacterial cell walls. Human cells do not have cell walls. Viral RNA molecules and viral and bacterial DNA. We also have our toll-like receptors or our TLR. These are innate immunity receptors that stimulate responses from the innate immunity cells. So let's cover the different innate immunity cells. So these cells these images are actually the representations of the anime for the specific cells. So we have our neutrophils. These are small motile phagocytic white blood cells. These contain granules and destructive chemicals. They are effective against bacteria and usually the first to arrive. So if you watch the anime later, you will notice that every time there is an infection, you will always see neutrophils that fight the intruder first. However, these neutrophils are unable to replenish granules and usually die by apoptosis. So when they die in sufficient amounts, they can generate pus, or in Tagalog, our nana. Next, we have our monocytes. So the lady on the right, you can see, these are our monocytes. Monocytes become macrophages when they enter the tissues, and in the anime, they only represent the macrophages. They are responsible for later cell stages of the infections. They clean up the neutrophil and cellular debris. They are found underneath surfaces of the body and around blood and lymphatic nodes. So these are actually ambush cells. Okay. So in the anime, the macrophages usually arrive later compared to our neutrophils. We also have our dendritic cells. This is the person on the left side. So in the anime, he is represented by a receptionist type character in which his hat has a branch attached to it. So this is actually related to the name dendritic cells. So dendritic cells, these are large motile cells which has long plasmic extensions. So the dendrite is actually an older term for branches. So this cell is represented in the anime as a person residing in a tree. So this actually reflects the cell's nature in which it is branch-like. It is concentrated around lymph nodes, has almost all innate immunity receptors, and it is extremely sensitive to pathogens. The primary function is to produce antigens that can present themselves to phagocytes. Next, we have our natural killer cells. So you will encounter this in the anime as a lady in a tank top. This recognize and kill host cells such as virus hosts and tumor cells. These cells do not exhibit memory, and the way they kill our cells is they induce apoptosis and promote macrophage phagocytosis. So in the anime, the natural killer cell is shown to kill cancer cells. Then we have our mast cells, basophils, and eosinophils. So these are granular, activated by antibodies to release these granules. When activated by antibodies, they become antigen specific, and antigens remain in these cells. Adaptive immunity is found in these innate cells. Okay, so we have our mast cell. So this is this is the representation of the anime as the mast cell. So these are non-motile cells. These defend the sites where they reside. They are found under the skin, lungs, and genitourinary tract. They can be stimulated to recruit neutrophils, eosinophils, basophils, and our T cells. This also releases inflammatory mediators, particularly histamine. So these are mast cells are notorious for eliciting an allergic response. So in the anime, the specific person is actually allergic to pollen, and you will encounter this person or this mast cells which elicits the allergic response of sneezing and inflammation of the airway tract. Next, you have your basophils. So these are recruited from the blood by the cytokines. And you have your eosinophils represented by the lady wearing a pink hat. So these are specific cells for parasites. Since they carry cytotoxins and neurotoxins, they are specialized to kill larger cells. So an increased number of eosinophils is characteristic of a chronic allergic inflammation, the main cause of damage in chronic asthma. Okay, so let's talk about the complement system. So the complement system is also part of our innate immunity, in which these are not cells. These are a group of plasma proteins that are found in the liver, produced in the liver, then circulate in the blood as non-functional proteins. So they become activated in a cascade. When we say a cascade, it means one part will activate another part. Okay. If you are familiar with our coagulation cascade in our blood, it is similar to our complement. So we have examples of cascade pathways. We have the alternate and lectin pathways. These are for innate immunity. For classical, it carries for both innate and adaptive immunity. So for the alternate pathway, it is the initial complement response. So in this pathway, C3 is the name of the complement. It is activated spontaneously in the blood. However, it is also deactivated immediately. So it pops on and off in the blood. However, if your C3 attaches to a surface of the pathogen, it is then stabilized. So how it works, it is like a random effect. So if a pathogen is nearby, it will stabilize. If it is not, it will deactivate. So it's a random roaming molecule. We also have inflammatory cytokines that can stimulate the liver to release mannose binding lectin and C-reactive protein. So mannose binding lectin, this is for the lectin pathway. It begins when mannose binding lectin binds to mannose on the surface of the pathogen. C-reactive protein binds to phosphocholine in the surface of pathogens and activates the C1 in the classical pathway. All these pathways will converge to produce activated C3 on the surface of the pathogen. And once the C3 is activated, these proteins can form a membrane attack complex. So the membrane attack complex is sort of like a facilitated hole. So here you go. So this is a diagram for our different pathways. It all leads up to C3, driving the rest of the pathway up to C9. And your membrane attack complex is actually this part here. So your membrane attack complex is like proteins that can tear through the cell membrane and cause a permanent channel to open. And this channel will facilitate the movement of ions and fluid into the cell, eventually killing the cell. For the innate protection against our intracellular pathogens, we have our interferons. So interferons are proteins that protect the body against viral infection. Viruses often stimulate infected cells to produce type 1 interferon. Dendritic cells can detect viruses and also produce type 1 interferon without being infected. Type 1 interferon receptors can be found in all human cells. And when neighboring cells detect type 1 interferon, it begins to produce antiviral proteins. Natural killer cells can recognize infected cells, damaged cells, and tumor cells, and when they detect our type 1 interferon, they can secrete type 2 interferon that can promote phagocytosis. Natural killer cells are the innate component of cytotoxic T cells. For our adaptive immunity, we have two general types. We have our antibody mediated, which is against extracellular pathogens. When we say extracellular, it does not invade our human cells. And results from B cell activation eventually to provide production of our antibodies. This is also called our humoral immunity. Cell mediated is against intracellular pathogens and results from the activity of our T cells, or particular our T helper and our T cytotoxic T cells. So before we discuss the other parts of our in adaptive immune system, we have to discuss first the major histocompatibility complex molecules, or MHC. So MHC are glycoproteins on the surface of cells that are involved with the activations of most lymphocytes. They bind to antigens and present it to T cell receptors. So their main role is to process antigens for it to be presented. We have two classes. We have class 1, which is found on all cells and presented in and present intracellular pathogens antigens to cytotoxic T cells. We have class 2, which is found on professional antigen presenting cells, or our APCs. This function to present antigens from extracellular pathogens. So MHC1 is for our intracellular, and MHC2 is for our extracellular pathogens. Professional antigen presenting cells, or are our APCs. These are our dendritic cells, macrophages, and B cells. APCs can take pathogens which include free viruses outside the cell. So we have our naive lymphocytes. So naive lymphocytes are our mature helper T cells, cytotoxic T cells, or B cells that is not yet exposed to the antigen which it is specific to. So if you watch the anime later, the naive lymphocyte is described or is represented by a young T cell, which is comparatively a lot more smaller than its mature counterparts. So antigens are brought from infected tissues to peripheral lymphatic tissues by our dendritic cells. So in the anime, the dendritic cell actually activates the naive T cell. There are two ways dendritic cells can present antigens, either by eating the pathogen and providing MHC2, or if the dendritic cell is infected by the cell, it can present MHC1. So antigens in the lymph, free-floating antigens in the lymph and blood, can also stimulate the macrophages and phagocytize them to display MHC2. So dendritic cells and macrophages expose naive T cells to their antigens, resulting in their activation. So first is the activation of helper T cells. So helper T cells assist in the activation of naive B cells and other T cells, particularly our cytotoxic T cells. So dendritic and macrophages phagocytize our pathogens. They then process the antigen complexes with MHC2, then present it to our naive helper cells. Only helper T cells with the T cell receptors that are specific to the MHC class 2 antigen complex can respond. To ensure that only helper T cells respond to the MHC class 2 antigen complex, so MHC class 2 antigen complex is simply the combination of the MHC class 2 and the antigen captured. A glycoprotein named CD4 must bind to MHC2. So CD4 is only found on our helper T cells. That is why helper T cells are also called our CD4 or our T4 cells. So when it complexes with our MHC antigen complex and CD4, the first signal is produced. However, this signal is not yet complete since you need a stronger signal to make the helper T cell activate. So a second signal is provided by co-stimulators. These are surface proteins on the APC. The most important one is B7. B7 binds with CD28 on T4 cells. B7 is on the APC, CD28 is on the T4 cells. Resting APCs have no B7. So resting APCs cannot elicit or awaken our helper T cells. So why is this process important? This is important so that the body does not waste resources producing helper T cells in which it has no purpose. A helper T cell responds to the antigen presented by the MHC2 molecules and co-stimulation. It then produces interleukin 2 receptors and interleukin. So this is one of those self-propagating processes. It both produces interleukin 2 together with interleukin 2 receptor. So the produced interleukin 2 will actually stimulate itself. So when it is stimulated by IL-2, it starts to divide and differentiate. Some helper T cells now become memory cells. What are memory cells? Memory cells are T cells that remember. That is why it's called memory cells. They remember the antigen. They are inactive. However, if they are stimulated by the same antigen, it rapidly becomes more helper T cells, resulting in an immune response. So this is the process of our memory or our adaptive immunity. Sometimes the immune response is so fast you do not know that you were infected. It's because your body was already prepared by preparing memory T cells. Helper T cells secrete cytokines that regulate other immune cells. Besides this, helper T cells can both awaken B cells and cytotoxic T cells. So that is why helper T cells are very much important. Okay, so to reiterate the whole process, the antigen will be absorbed by an APC. Next, the APC will merge the antigen together with the MHC class II molecule, forming the MHC2 antigen complex. It will now be attached to the T cell receptor present on our helper T cell. Now, to verify, the helper T cell will attach CD4 to MHC class II, and this is the first signal. The second signal is facilitated by attaching B7 and CD28, and when they bond together, this forms our second signal, or our co-stimulation. Once the co-stimulation occurs, the helper T cell becomes active. When it is activated, it will release interleukin 2 and it will form interleukin 2 receptors. Interleukin 2 will activate itself and it will start the helper T cell to differentiate and divide. It will form more helper T cells and some will become memory T cells. Next we have is for our antibody mediated immunity, the B cell receptor can bind some antigens without the MHC molecule. However, it is relatively weak compared when the B cells are activated by helper T cells. Most B cells do not respond to antigens without proper stimulation from helper T cells. So the process for activation of our B cells is, first, B cells take in the antigen. This is optional. Okay. So the antigen is processed and presented as MHC2 antigen. Then this T cell receptor of an activated helper T cell binds to MHC2 antigen and CD4 binds again to MHC. Helper T cells secrete interleukin 4. Okay. Interleukin 4 stimulates the B cells. When the B cells are now stimulated, it will start to divide and differentiate. Some will become memory B cells, while some become plasma cells that will produce our antibodies. So this is the process. Okay. So the unprocessed antigen will bind to the B cell receptor, and the helper T cell can also present the antigen with our MHC2 complex to our B cell. Okay, so when the B cell is has bound successfully, the helper T cell will now release interleukin 4 that will activate our B cell. Our B cell will now start to divide and differentiate. Some will become memory B cells, while some will become plasma cells, will eventually produce our antibodies. So next we have the function of cytotoxic T cells and cell-mediated immunity. So cell-mediated immunity is particular for our intracellular organisms. So natural killer cells kill viruses infected cells during early stages of a viral infection, while cytotoxic T cells handle the later stages. So in activation of cytotoxic T cells, only dendritic cells can provide enough stimulation to activate naive cytotoxic T cells. Dendritic cells acquire the antigen either by phagocytosis or being infected. The antigen is processed and MHC1 is presented to the naive cytotoxic T cell. Only cytotoxic T cells specific to MHC class 1 antigen complex responds. CD8 now binds with MHC1 molecule. So in our B cells and our helper cells, it was CD4. For our T cells, it is CD8. So cytotoxic T cells are actually called your CD8 or T8 cells. So co-stimulation occurs when B7 binds to CD28, like in our previous activations. And cytotoxic T cells produces IL-2, and IL-2 makes T cells divide and differentiate. If you remember, IL-2 is also produced by our T helper cells. So small percentage of the T cells become memory T cells, and when stimulation by the dendritic cells are insufficient, helper T cells can provide assistance. So this is how it works. There are two ways. It's either the helper T cells will provide the interleukin 2, or the cytotoxic T cell will provide interleukin 2 to itself. So the APC will present the antigen will be bound to the MHC class 1 molecule antigen complex. It will bind to the T cell receptor found on the cytotoxic T cell. Then the T cell will become activated. If it is activated, it will produce interleukin 2, which is bound to itself, and it will cause division of our cytotoxic T cell. Some T cells will become memory T cells, while some will become daughter cells of cytotoxic T cells. And these cytotoxic T cells can kill our infected cells. If this is not available, helper T cells can provide interleukin 2 since they can also produce interleukin 2 to our cytotoxic T cells, enabling them to also be activated. So the effect of our cytotoxic T cells is it can recognize the cell to the T cell receptor. It can detect the antigen present on the cell through MHC1, and because of specificity, it can only attack infected cells. Cytotoxic T cells release perforin, and it forms a channel in the plasma membrane. Granzymes can enter the channel and activate the enzyme in the cells to undergo apoptosis or programmed cell death. So that is all for the special lecture on immunity. I hope you have learned something, and I really suggest you watch the anime. It's really good, especially since you're going to be future medical professionals. You can relate to that anime. Thank you and good day.